Yeah, they opened up one on Ann Arbor State.
Oh, did they? Okay.
There's got to be a limit. The Meyer there,
Okay.
built outbuildings there, there's one there.
Right, so more interstate traffic,
Yeah.
which makes sense. People driving through with money as opposed to college students with no money.
You brought your sunglasses.
Well, just the rent. I mean, everybody's losing because the rent is so high down there.
So, good afternoon, everyone. So today's Tools and Tech will be conducted by Tony, a student in my lab. Tony, how long have you been here? About 2 years, maybe? I don't even know. He shows up every day. He's at the desk. I'm like, oh, hi, Tony. Just showed up one day. He was like, okay, here's a laptop. Do good stuff. So Tony's been working on a number of different projects with, in part with a collaborator over in the neurosciences, Peter Todd and his group. And we're looking at the role of and the prevalence of ribosomes on different transcripts and how we can model that and measure that. And as part of this work, Tony has developed a tool that he'll talk about today for different ways to measure and actually identify regions of active translation. So take it away, Sam.
So the talk I'm going to give is on an algorithm that we developed called SPECtre. It's a spectral coherence-based classifier that looks at actively translated transcripts in ribosome profiling data. Other than the algorithm, what Ryan told me in developing this is the second most important thing to have is a cool name. So, hence SPECtre. So just as an overview, I'll give some background, and then look at a couple of the other classification algorithms that exist right now. And speaking of cool names, ORF4, highly imaginative, is one of the other algorithms that's out there. And then I'll walk you through the pipeline as well as looking at some test data and how the different classifiers perform using that data. So we've traditionally understood the transmission of information from DNA to RNA to protein to be linear. What we've come to realize recently is that isn't exactly so. We have multiple mechanisms of regulation that ultimately can alter how DNA and RNA and protein are expressed. So I've just listed a few of the major mechanisms that are in play here. But what we see here is that again that going from DNA to RNA to protein is not always linear, and what I'm going to focus on today is looking at how RNA is translated into protein. So to give some perspective on some of the techniques that are commonly used to measure the output or the activity of a cell to transcribe and then ultimately translate RNA to protein. We have RNA and mRNA-seq to look at kind of the total transcriptional content of a cell or group of cells. BREW-seq, which is a technique developed by the Youngman lab here at Michigan, looks more at the nascent active transcription of a cell. And then ultimately you can use mass spectrometry among other techniques to look at protein abundance. But what I'm focused on today is a fairly recently technique developed called ribosome profiling, and that looks more at the translational content of a cell or group of cells rather than the transcription. So just as a brief overview of next generation sequencing, you have your RNA here, which is either fragmented to get total RNA, or you can do some sort of enrichment using like an oligo DT to get mRNA, to enrich your mRNA. Those RNAs are then fragmented, and then adapters and sequencing primers are ligated to the ends of those. Those are then loaded onto a sequencing machine, and usually, I think in the major method right now, sequencing by synthesis, single nucleotides that are labeled are added to the flow cell, and their incorporation will give off a signal, and the machine will read the sequence that's been synthesized. Once you have these sequence reads from the machine, you'll do some QC and align those to a reference genome or transcriptome. And that will enable downstream analyses like expression profile and things like that. So to speak a little bit more about alignment, again, we have these RNA reads. And there are usually two major methods of alignment. The first is either de novo, so that's basically assembling transcripts from the reads themselves and then aligning those transcripts to the genome. What most people do with mRNA-seq or in ribosome profiling is to align those reads to a genome and then assemble those transcripts and then look for differences and then the abundance of reads over different splice models can help you estimate which isoforms of genes are more or less abundant. So to speak a little bit more about ribosome profiling, because not many people are as familiar with this as with RNA-seq. You start just like with RNA-seq with a cell. You lyso cells and use either a sucrose cushion or gradient to isolate polysomes versus ribosome or monosomal fractions of ribosomes bound to mRNA. Nucleus digestion will then yield fragments of mRNA with their bound ribosomes. And then what usually happens at this step is that further enzymatic digestion will release those mRNAs further enzymatic digestion and denaturation will release those mRNAs from ribosomes that were previously protected by the ribosomes. And then from there on, linker ligation and cDNA synthesis proceeds just like any other RNA-seq experiment. Just briefly, there's one of the major differentiating points about ribosome profiling is that your cells are usually treated with different inhibitors, translational inhibitors. The first two, I believe, act similarly. Herringtonine, I think, what does it do? It will bind to free 60S subunits in the cell and will prevent basically elongation once it's formed an 80S subunit. It won't prevent elongation of ribosomes that are bound prior to treatment. So there has been in the literature some observation that you get a little runoff. It's not perfectly at the initiation sites. The other two also, I believe, act similar to each other. They provide kind of a more uniformly distributed view of translation. Cyclohexamide specifically, I believe, changes the conformation of the E site in the ribosome such that it interferes with the tRNA transfer or something to basically prevents any further elongation. So just to review, herringtonine and lactodomycin, they will generally stall ribosomes at initiation sites. Cyclohexamide and emetine will kind of stall ribosomes wherever they're found. So there are a couple techniques out there that look at sequencing from the standpoint of proteins that are bound to them. So I just wanted to briefly review those within the context of mRNA-seq. So one of those techniques, it's called ClipSeq. It stands for the cross-linking immunoprecipitation followed by sequencing. It also goes by some other names, parclip, parcle, or something like that. But the general idea is that you're also isolating proteins that are bound to mRNA, digesting, and then sequencing the bound fragments of mRNA. So with ClipSeq, you can basically look for different proteins. I think the Kin lab that was here at U of M, they developed a more generalized ribosome or RNA binding protein protocol, but usually it's a specific protein. Riboseq obviously will isolate mRNA bound by ribosomes. The arrest method used in methods like ClipSeq is usually cross-linking or some sort of chemical cross-linking procedure like formaldehyde. Riboseq, like I mentioned, uses those translational inhibitors. The method of recovering those mRNA RBP complexes are both similar across the two methods. mRNA-seq obviously does not require that. The major difference between ClipSeq and those methods and Riboseq is that with ClipSeq you can still do an oligo-DT enrichment. With Riboseq you can't. And because of that, I'll go into a little bit more detail why that's an issue. And then nuclease digestion across those two methods are the same. In all three cases, you can do a ribosomal RNA depletion step to kind of cut down on the contamination from those sources. And with mRNA-seq, you can obviously do single or paired end. ClipSeq and Riboseq are limited to single end sequencing. So there are some inherent advantages and disadvantages, like I mentioned with ribosome profiling. The first is that there's a high background of ribosomal RNA, and the reason this is that the RNA's digestion will target not just the unprotected mRNA, but also the ribosomal, unprotected RNAs, but also the what's found in the ribosomal complex itself. So therefore, this leads to RNA contamination being dependent on the efficiency of your depletion step. And scary enough, some ribosome profiling experiments that I've seen can be comprised of up to 80% rRNA contamination, but usually 10 to 40% is more typical. Because of this, sequencing of your target mRNA competes with that of rRNA, and so your sequencing depth and diversity is usually much lower than you will see with mRNA-seq. Higher coverage libraries that I've seen published may have like 100, 150 million reads. And obviously multiplexing with ribosomal profiling because of these concerns is an issue. But one of the neat things about ribosome profiling is that when you align your reads to a reference transcriptome, the alignment of those reads, the position of those reads actually tracks the 3 nucleotide periodicity of a ribosome as it translates transcript from RNA to protein. And the reason this is, well, it's not a bug, it's a feature. But the reason this is as your mRNA is being translated, it's being added 3 nucleotides at a time. And once those are added, it moves in a 3 nucleotide window. And so when you align those reads to a reference transcriptome and look at the coverage over a transcript, look at the normalized coverage over a transcript, you'll see that there's a clear a peak every 3 nucleotides, with good regularity. So in black here, that's ribosome profiling. When you do the same thing with mRNA seq library reads aligned to the transcriptome, you don't see that same periodicity signal. And so this idea forms the basis of our algorithm. So what we're interested in as a lab, in concert with our collaborators, is looking at upstream open reading frames. And so why this is important and how this algorithm plays into it is that it's often very hard to tell what's actually being translated versus what isn't. And so just to give a brief background about UARFs, approximately half of the human transcriptome contains a UARF. Many of them actually contain more than one. And this mechanism of upstream translation is conserved across species. And so the reason why these UARFs are important and their expression is important is that they may actually negatively regulate protein expression. 2 major mechanisms through which it does that is through the promotion of mRNA decay. As it's read here, there might be as it's read from this upstream frame into the mRNA, it may introduce like a non-canonical stop or something like that, and trigger the nonsense mediated decay pathway. The other major mechanism is just through competitive expression. If this is being translated by the translational machinery at the expense of this one, you'll have high expression of this one versus your canonical protein. And so these UARFs have been found in genes with a diverse array of functions. They've been found in oncogenes, genes related to differentiation, which is what we're ultimately interested in, cell cycle control, as well as stress response.
So when they have them fully upstream, do they divide them up into, or distance between the stop codon for the upstream and the start codon for the...
Whether they differentiate between whether that's like a novel protein or...
No, as to expression level of the downstream thing.
Oh, yeah. So the distance will have an effect.
Yeah.
Sometimes if there's sufficient distance for the ribosomal, for the translational machinery to basically pick back up, it's unaffected.
It was the other way, that the stop has to be close.
Right. If
because it will not reinitialize it.
Right. You're right. Yeah. If they're close together, then right, it won't be able to.
It doesn't disassemble. It just picks up.
Yeah.
Yep.
Is there any role of like codon usage in this particular like starting code? Because like there was one paper published in Cell, they say like there is RAM, the starting, so like starting 20 or 20 to 30 codon, they use like rare codons. So like from the starting, translation machine they start very slow, then with time they pick up.
Yeah. So there is an aspect to that, and one of the things that so I think another thing that our lab is also looking at is if you have basically a site that is canonically not an AUG, but that doesn't actually answer. But another thing that we're looking at is looking whether a mutation can introduce an AUG, but codon usage. We get back to it in a second. Yeah. So because of this, it's these aberrant expression of these UARFs have been implicated in a number of diseases, and including cancer, pancreatitis. So just to give a little more background on some of the existing methods that are out there already, the aforementioned highly imaginatively named ORF score basically takes the alignment of reads in each frame and compares it against a uniform distribution and computes a score based on that. So if there is enrichment of reads in a non-canonical frame, it will penalize that, which is, I think, part of the reason why it doesn't perform as well in the classification. The other method that was published by the lab that initially developed ribosome profiling is called Floss, which stands for Fragment Length Organization Similarity Score. And basically it looks at the magnitude difference at the distribution of read lengths in protein coding transcripts as a reference signal here in red versus your transcript of interest, and based on that magnitude difference, it will either qualify or not as a protein coding transcript. So most non-coding RNAs and mitochondrial transcripts have a distribution profile that's distinct from protein coding transcripts here in the darker blue here. Long non-coding RNAs and 5' UTRs of protein coding genes actually follow the profile set by protein coding genes. And so this was actually, this comparison here was the basis of their paper where they posit that that there may be pervasive translation of link RNAs in the genome.
Are they defined as non-coding? Like,
How are they defined as non-coding? So it's based on annotation. So what they did was they took basically anything that wasn't a pseudogene, anything that wasn't a link RNA, and basically lumped those together, and they also and that gave them a set of non-coding transcripts. They then added a further filter where anything that overlapped with a protein coding gene could not be considered non-coding. And anything within like a certain proximal distance, I think they used 5,000 nucleotides as a filter too. So basically they used a sanitized set that they had annotated as non-coding. So for the classification test that I did, I actually followed the protocol set out by them just to make sure that the comparison was one-to-one. So the way that they do their classification is that they first plotted the Floss scores of every transcript as a function of the number of reads aligned to those transcripts, and then used a rolling window of 200 transcripts starting from the lowest number of reads up, and calculated cutoff using an extreme outlier cutoff. And so that's shown here. So you have the Floss scores here, the number of reads on the x-axis, and they used this, I believe it was a lowest smoothing curve to define their cutoff. One issue that I would like to point out, in which we were kind of surprised by, is what can you actually say about transcripts that only have 1 or 2 or 3 reads. When I pointed it out to Brian, we were actually kind of surprised this got accepted. But nonetheless, this is their idea is that as long as their Floss score as a function of their input read followed a certain distribution, they called it a protein coding gene. And their published accuracy using this method they claimed they were able to recover 99.6% of protein coding transcripts. So I'll get to this a little bit more later, but just to give an idea of the algorithm that we developed, it's based on spectral coherence. So coherence is, trying to keep this a little bit more high level. It's basically the relationship as a function of frequency between two data series. And so this is kind of, this is a simplified example here, but if you have two waves here, two sine waves here, one, the first one here, X, is a sine wave with a frequency of 10 hertz. And if you have a second wave here with, that's composed of two signals, actually, one at 10 hertz and one at 27 hertz. If you look at the spectral power spectrum of each of those waves, you'll see that you have a peak here for wave X that corresponds to 10 hertz. For wave Y, you see two major peaks at 10 hertz and 27 hertz. When you plot the coherence function between those two waves, the only wave in common between the two, the only signal in common between the two is, of course, 10 hertz. And so you see this high, what's called magnitude squared coherence at 10 hertz. So the idea that we're working off of is that regions of active translation will have high coherence with a signal associated with that trinucleotide periodicity signal. Didn't make it. Okay. And what we do see, I think I'm missing a plot here, but I had a plot here where if you look at the distribution of SPECtre scores for protein coding transcripts and for non-coding transcripts, we see a nice delineation between the two distributions. So to walk you through schematically what we're doing, for each transcript, we generate a normalized, we calculate the normalized read coverage signal based on, you have to do an adjustment of the 5' end position of the aligned reads because the reads that are sequenced are protected by ribosomes and the A site or P site is in the middle of them. So you have to adjust for that positioning aspect. So we generate this normalized read coverage signal over transcript. And then we also generate in tandem an idealized coding signal that corresponds to that 3 nucleotide periodicity, like so. We then calculate the coherence between this normalized signal and the reference signal here. And then
Kind of predicted that might happen.
So do you get your idealized coding signal, you just made it up, or you get it from the data itself, of the coding?
So we've done it both ways. And the problem with, well, so one of the reasons why we got away from taking it from the data itself is kind of one of the inherent problems of the Floss algorithm too, is that the Floss algorithm, it's highly dependent on the read distribution you get following your size selection. And so we kind of just wanted to look at it like if we had this ideal signal. And given that spectral coherence, it's really only concerned about the frequency aspect of your signal. It kind of really didn't matter too much to us. But yeah.
Not to calculate coherence by cross correlation or what?
No, it's coherence, but it's related to cross correlation. Unlike cross correlation takes into account the magnitude of the signal as well. For coherence, we're only concerned about the frequency, how two signals correlate by frequency. Yeah, so what we then get is the coherent signal across various frequencies that are tested. And what we extract then is the coherence at the, this is plotted as the inverse frequency, so 0.33 corresponds to that 3 nucleotide periodicity frequency. So regions with perfectly inactive translation should be 0, very low should be skewed towards 0. Higher translational activity should tend higher to or closer to 1. Oh, well, there's the distribution. So the way we implemented this algorithm is that we enabled it so that you could measure coherence over either an entire transcript or over sliding windows across a transcript, like so. Take the coherence there, and so on. And then what we did for the purposes of this, for the test data, is we took the median coherence of these windows. The program itself, you can direct it to take different metrics as well. So for the comparison of the different classifiers using the test data, we looked not only at the entire transcript, but we also looked at 30, 60 nucleotide windows. And I'll mention why we did that briefly. So just to give an overall view of how we processed the how this data was processed. After trimming adapters and aligning to an rRNA contaminant database, you take the unmapped reads and align those to the reference genome and transcriptome. We filtered based on mapping quality, quantitative transcript abundance, and the script itself will be able to support different input formats for that, and then calculated the SPECtre, Floss, and OR score for each transcript. So we implemented the we implemented this in Python with an eye towards trying to cut down on extraneous third libraries. So most of these are actually default libraries. The analysis itself requires a native installation of SAM tools as well as R with the ROCR package, and then to do the calculations themselves, I do those using RPy2. So what we take is first feeding in the transcript GTF, we load all the transcript coordinates into memory in the form of an interval tree. We then load the BAM alignments into memory, and after adjustment to take into account that A site or P site position within the ribosome, that's also converted into an interval tree. I then removed overlapping transcripts as the Floss experiment did, where they removed overlapping transcripts as well as any transcripts within 5,000 nucleotides of each other. I then extracted the coverage over each transcript, and then calculated the SPECtre, Floss, and OR scores for them. Those scores are then used to build a distribution, for example, coding versus non-coding, or translated versus actively translated, depending on, say, like an arbitrary FPM cutoff. And from that, I can calculate experiment level statistics. And so the plan, so right now we're able to output transcript level metrics for each transcript in the GTF that is tested, experiment level metrics as well as plots. So a quick word about the data itself. I used the Floss, the data that was used for that Floss publication, which were mouse embryonic stem cells treated with cyclohexamide or emetine. Since the rest of these were treated with cyclohexamide, I used only the cyclohexamide library. The second library or data set that I used for this test is from another 2014 publication looking at ribosome profiling in zebrafish, and so this group actually are collaborators of this group. So just keep that in mind for later. And so for their meta-analysis, which I copied for this test, they combined all 16 of their ribosome profiling libraries into one for a meta-analysis. This amounted to almost 500 million reads. And then our collaborators, as part of a ramp-up test for a larger project, did a test library in SY5Y neuroblastoma cells. So I tested these using using a binary classification. First, I did either whether it was as I had done with that Floss publication, whether it was annotated as protein coding or non-coding. We also looked at various FPKM cutoffs to basically differentiate whether it was actively translated or not actively translated. So just a quick word about RFC curves. So given a cutoff point in a binary classification test, you calculate the true positive rate versus a false positive rate. So for a test with perfect sensitivity and specificity, an ROC curve based on that would basically cut through the top left portion of this graph. The closer your curve is to the diagonal line, basically that indicates you might as well flip a coin. And so based on these ROC curves, you can also calculate the AUC, which is the probability that you will rank a randomly chosen positive instance higher than a randomly chosen negative one. So that can give you a measure of performance as well. So what I did was to, in the Golia mouse embryonic stem cell data set, computed the Floss or score and SPECtre scores using the methods that they had previously published. And then, like I mentioned previously, looked both at looked at various ranges. So starting here at 1 read, which is what they did. What we have here in the solid line is the AUC. In solid lines are the AUC at this point, using this point as the minimum cutoff. And so here in solid black is the ROC based on Floss, and in dashed line is their published accuracy. And so what we see here is that using a couple of windows, particularly if you notice the 30 nucleotide window, it outperforms their classification algorithm once you impose a cutoff to differentiate active versus inactive translation. And so keeping in mind that this is the lab that developed, published, and have published many, many papers on ribosome profiling, the fact that we're beating them at their own, obviously, or most likely cherry-picked set of data was good for our confidence, at least. So yeah, and so obviously, and so one thing you'll also notice that here in purple is looking at the spectral coherence score using the whole transcript. And the reason why looking at the whole transcript doesn't work as well as looking at these windows is that, as I'll go through later, spectral coherence is based on a signal. And so if you have gaps in read coverage and there's no signal there, it's unable to basically formulate a score. And so we found that windowing, using a windowed approach does seem to improve performance quite a bit. Oh, and then at the bottom is OR score. So the second set of data that we looked at was the Vizzini zebrafish data. This was yeah. So this group is our collaborators of the Weissman lab in Nick and Golia, and so again we see that in this mostly independent data set, we see that all three windowed approaches outperform Floss as well as OR score.
So did you try an even smaller window size?
I
Since your
haven't yet.
smallest is best.
I haven't
Or
yet.
push it farther.
Yeah. So looking at the data generated by the Todd group here at Michigan, it was prepared by his student Caitlin, and this was like I mentioned before, a test library for a larger scale project, which I'll talk about next week, I'm pretty sure. There's again, our windowed approach outperforms Floss, and the reason why we think that why Floss seems to perform worse and worse as you look at as you go further and further away from their own lab, is that Floss because it's based on that based on scoring using those read length distribution profiles. It's going to be sensitive to any variation in that, and so that kind of leads to whether or not the degree of reproducibility from experiment to experiment, much less from lab to lab. And so yeah, so like I mentioned, its inherent advantage is that it's very simple, and in most cases it actually does perform quite well because it's based on this expected fragment size distribution. But because of that, it's also sensitive to variations in the efficiency of fragment size selection. And so what we have here in black is the Golia data, which follows a certain profile. When you look at the Vizzini lab data, it follows slightly similar, but obviously quite different read length distribution profile. And then the Todd data itself follows an entirely distinct distribution itself. The published accuracy correctly classified, 99.6% of protein coding genes from non-coding RNAs. But like I mentioned before, that published accuracy might be a bit overstated due to the functional insignificance of these transcripts with low read coverage. Unlike SPECtre, however, it is robust to gaps in read coverage because you're looking at an overall read length distribution as opposed to a signal. For OR score, it also likewise is robust to gaps in read coverage since it's looking at an overall read distribution by frame. One issue may be that it's overly punitive to transcripts that demonstrate translation outside of an annotated reading frame, which may affect its classification performance. What I think is an advantage of our algorithm is that it is signal-based, so therefore it seems more robust to technical variations in that fragment size selection process. But like I mentioned previously, in comparison to using a cross-correlation metric, it's amplitude agnostic, meaning that its input and output coherence is a function only of its frequency and not its magnitude. It seems to be quite accurate, especially when you look, at windowed, especially when you start breaking it down into windows across a transcript, and more so when you start looking at defined points of what is considered actively translated and those that aren't. And like I mentioned, it's sensitive to gaps in read coverage, since ribosome profiling coverage isn't always uniform or robust across all transcripts equally. The other advantage that we have is that we're offering this as a standalone package. Neither Floss or Score are provided as package binaries. In fact, it was recapitulating Floss was in and of itself a huge exercise. First of all,
They don't distribute code for it?
they have code, but they don't provide the annotation files. They don't provide the code itself is not commented very well, so it was actually really hard to follow. And so actually, just to mention, I did speak with Nick and Golia over email a couple times, and he was actually really helpful. But yeah, it definitely was a process just to even recapitulate their analysis. What was actually even more frustrating, despite how helpful he was, is that he's decided to migrate all of his code and any other improvements that he's making post-publishing of that paper into this code. The original code that I was trying to recapitulate from was from R. He's decided to convert everything to Haskell. Yeah. So yeah. So I'll just briefly mention the outputs that we'll have from our program is we'll have these transcript level metrics, experimental metrics, as well as plots, both for the coherence over a transcript, those distributions. And one thing we're actually actively working on is based on either these windowed coherences. This is just a toy example here, but based on these windowed coherences over a region, or even using the posterior probabilities across a region, we're actively looking into basically implementing like an HMM to define where we might see the start of active translation versus not. And that can help us pinpoint where these upstream open reading frames might initiate. So it's very close. It's not up quite yet, but this is where you can find everything. Yeah. So I'll just briefly acknowledge my lab here, our collaborators in the Todd lab, and Caitlin Rodriguez here, and then my funding sources there.
Did you try any other simple algorithms before you settled on the coherence score?
I did. I tried, like I mentioned, I tried cross correlation as one of the initial tests. Just because of the variances in signal, it was hard to get a normalized, idealized signal. That basically the scores were, it was really messy using that. I even tried, and along the same lines, I tried just a straight Pearson correlation. That didn't work too well either.
Yeah, I was thinking more along the lines of like the ratio of first base pair to the second to the third, and the third to then
Oh,
the
yeah.
first to the next one. What you would expect is a high value, about
Mm.
1, and then a low value, and
Yeah.
then that would repeat over and over.
Yeah.
Did you try fast Fourier transforms on the data?
So the coherence itself is based on the Fourier transform of the data. The math still eludes me, but yeah.
And I noticed, or at least you don't mention it, doing some of it in NumPy.
I thought about doing that.
Some of that's a lot faster.
Yeah. It would be, but at the same time, I also kind of wanted to make it so that there wouldn't be. No, I mean it's something that I'll probably revisit. But yeah, it was something that I, at least for this iteration, I was trying to do with as much standard library as possible. But yeah, no, that's definitely something I'll probably revisit later versions.
I got a teacher.
Okay. Thank you.